Hi, welcome back. In this video, we will discuss about cell division and we will focus on mitosis. Describe the four stages of mitosis and the behavior of the chromosome for each stage. Mitosis is a type of nuclear division which produces two daughter cells, each having the same number of chromosomes as the parent cell. Before we discuss more about mitosis, let's understand a few terminologies that is important to explain about chromosome during mitosis. First, chromatin. Chromatin is the complex of DNA and proteins that makes up the eukaryotic chromosome. When the cell is not dividing, chromatin exists in a dispersed form as a mass of very long, thin fibers that are not visible under the light microscope. Chromosomes is a highly compacted structure from condensed chromatin, a cellular structure carrying genetic material found in the nucleus of eukaryotic cells. It consists of one very long DNA molecule and associated proteins. Now we will look into this diagram and we will try to understand the relation between the DNA, chromatin and chromosome. DNA double helix consists of a long polynucleotide and chromatin consists of elongated DNA. Chromatin that's become associated with histone protein will be called chromosome. The chromosome can become a condensed chromosome to become sister chromatid. Mitosis consists of prophase, prometaphase, metaphase, anaphase, and telophase. Based on our syllabus, the four stages of mitosis, it consists of prophase, metaphase, anaphase, and telophase. To explain about the behavior of chromosome during mitosis, we can focus on the three components. Number one, chromosome. The chromosome may be in the form of sister chromatid or in the form of chromatin. Number two, the centrosome. The centrosome may be in the form of ester, spindle fiber, or microtubule. And number three, we will explain about the nuclear envelope, whether the nuclear envelope disappear or disintegrate or break into fragments, and the nuclear envelope reform. Now let's focus on prophase. During prophase, the centrosome begin to migrate to the opposite poles and the chromosome already become condensed to form sister chromatid. During prophase, the chromatin fibers become more tightly coiled, chromosome condense and they will become shortened and thickened. Each chromosome consists of two sister chromatids joined at the centromere. Mitotic spindle or spindle fibers form or extend or elongate from the centrosome. In animal cells, ester will radiate from centrioles within the centrosome. The nuclear envelope and nucleolus disappear, centrosome move to the opposite poles. The next stage is prometaphase. Remember, in our syllabus, prometaphase is not the main stages in mitosis. However, let's take a look into events that occurred during prometaphase. During prometaphase, the nuclear envelope breaks into fragments. The microtubules extending from its centrosome and can now invade the nuclear area. The chromosomes have become even more condensed. A kinetochore, a specialized protein structure, has now formed at the centromere of its chromatid. Some of the microtubules attach to the kinetochore to form kinetochore microtubule, which jerk or move the chromosome back and forth. Some non-kinetochore microtubule will interact or overlap with the other spindle fiber from the opposite poles that will lengthening the cell. Now we will proceed to the next stages of the mitosis, the metaphase. During metaphase, the spindle fibers fully develops. The centrosomes are now at opposite end of the cell. The nuclear membranes disappears completely. The chromosomes arrange themselves or align or assemble at the metaphase plate of the spindle fibers. They are attached to certain spindle fibers by a protein called kinetochore. Some of the microtubules attach to the kinetochore to form kinetochore microtubule, which jerk or move the chromosome back and forth. The non-kinetochore microtubules interact or overlap with those from the opposite pole of the spindle that will lengthening the cell. The next stage is anaphase. Anaphase is the shortest stage of mitosis, lasting only few minutes. The centromeres divide or split, and the shortening of the spindle fiber will pull and separate the sister chromatids. Each sister chromatid is now pulled or moved towards the pole. Because the microtubules are attached at the centromere region, the chromosome moves centromere first. The energy for this process is provided by mitochondria. Chromatids that separate are now called chromosome. The cell elongates as the non-kinetochore microtubules lengthen. The chromosome moves centromere first. The last stage of the mitosis is telophase. During telophase, the chromosome or chromatid reach their poles or assemble at opposite poles. A new nuclear envelope forms around each group. The chromosome uncoil and lengthen, thus becoming invisible again. The spindle fibers disintegrate. The nucleolus reform in its new nucleus. The division of one nucleus into two genetically identical nuclei is now complete. Mitosis is followed by cytokinesis. Cytokinesis is the division of the cytoplasm to form two separate daughter cells immediately after mitosis. In animal cell, the cytokinesis occurs by a process known as cleavage per row. The cell membrane is pulled inwards by the cytoskeleton that consists of actin and myosin contractile ring. A shallow groove is formed in the cell surface near the old metaphase plate. A contractile ring forms on the cytoplasmic side of the furrow. The microfilaments consist of actin and myosin begins to contract. That will reduce the diameter of the ring until it slices all the way into the center of the cell. The cells are completely separated and the two daughter cells are formed, each with its own nucleus and its own share of cytosol, organelles, and other subcellular structures. The cytokinesis in plant cell occurs at the end of the telophase. The vesicle derived from the Golgi apparatus move along the microtubule to the middle of the cell, where they fuse together, producing a cell plate. The cell wall materials carried in the vesicles collect inside the cell plate as it grows. The cell plate enlarges until its surrounding membrane fuses with the plasma membrane along the perimeter of the cell. Two daughter cells result, each with its own plasma membrane. A new cell wall arising from the contents of the cell plate forms between the daughter cell. To conclude, the cytokinesis in animal cell is through the formation of cleavage furrow, and the cytokinesis in plant cell is through the formation of cell plate. The comparisons between the cell division in animal cell and plant cell. In animal cell, the cell division will occur in somatic cells throughout the body, while in plant cell, it occurs mainly in meristem that consists of the shoot and root tips of the plant. In animal cell, the cell division will involve the presence of centrioles, while in plant cell, there will be no centriole. Instead, the plant will have the structure with similar function that called microtubule organizing center. In animal cell, it will involve the ester formation, but in plant cell, no ester formation. In animal cell, the cytokinesis occurs by the constriction of the microfilament that will be called cleavage furrow, while in the plant cell, it occurs by the growth of the cell plate through the fusion of vesicle derived from the Golgi apparatus. The similarities of the cell division in animal and plant cell. Both will involve four phases, consist of prophase, metaphase, anaphase, and telophase. Both will occur in the somatic cells, and both will produce two identical or similar daughter cells. In plant cell, the cell division will involve no centriole, no ester, and there will be a presence of cell wall and also will involve the formation of cell plate during cytokinesis. This is the example of mitosis in plant cell that consists of prophase, prometaphase, metaphase, anaphase, and telophase. So that's all for now. Thank you and see you again in the next video.